We're standing behind the I Hotel here in the Research Park of the University of Illinois. There's just an area back here that hasn't been developed. This is what we call a bug vac. It's basically just a leaf blower with a vacuum attachment, and it's got a net bag taped to the end. So now we just empty out this net bag, and basically the way this works is that there's a little hole in here, and so the insects are going to be attracted to light, so they're going to come up out of all the debris and duff that's down in the bottom of this, and they're going to come out into this clear bottle. There's some baby grasshoppers, some crickets, there are a lot of little flies. There are some little tiny parasitic wasps. This is pretty typical of what you find in a sort of a wet grassland like this. Basically, there are a lot of different things you can do with the specimens in a collection like ours after you get them out of the field and prepare them properly and put them in the collection. So there are things like you can study their anatomy, you can extract DNA from them, you can also look at their gut contents to figure out what they were eating. One of the really hot topics in insect research right now is pollination biology, and that's sort of the study of which insects pollinate which plants, because insects are some of the primary pollinators of plants, and they're sort of critical to plant reproduction. One of the things you can do with collections is you can go in and actually get pollen off of the individual insect specimens, and there are actually keys that you can use to identify that pollen and figure out what plant it came from. One of the really useful things about collections and having that data from the collections available via InvertNet will be the ability to actually look back into the past, because a lot of these collections were started back in the 1800s. And so we actually have specimens that were collected more than 150 years ago. One of the really important uses of insects in environmental sciences is what we call bioindicators. What that means is, instead of going out and measuring something like the quantity of some pollutant in a stream, you can actually get that same information by looking at the species of insects that are in that stream, because certain species are going to be very sensitive and others are going to be less sensitive. And so if the sensitive species have all disappeared, then you know something's wrong. And as you know, many of the streams and rivers in Illinois have been severely impacted by agriculture. So we can actually look back and see that back before all these changes happened, there were a lot more species in those streams and rivers than there are now. And if we want to try to do restoration work on those streams, we can figure out, okay, which species do we need to try to reintroduce to those areas to reconstruct the native biota of that area. So InvertNet will serve two purposes in terms of research. One is the sort of the basic side, which is reconstructing the tree of life. But then there's also the applied side, to actually be able to go back in time. Now, we can't physically go back in time, obviously, but because we have collections that represent the biota of various areas in the Midwest that go back all the way back 150 years, we can actually get all this information off of these specimens, including when they were collected, where, what species occurred in particular areas at particular times. And we can sort of take all that information and combine it to give us a pretty clear picture of how the biota of that particular area has changed over time. This will be a very powerful tool for studying things like climate change and the effects of human activities on the natural world.